This is a little crash course on mitosis. What is it? A great example on the importance of mitosis is the amount of cells it replenishes on our skin. We lose an average of 40,000 skin cells an hour. How does it work? Another example is how a lizard grows its tail back through cell regeneration. This is the interphase, pre-mitosis. In this step, centrosomes are duplicated and two sister chromatids are made. The first step is prophase, which many would consider early prophase. As you can see in the sketch here, there's two centrosomes and the nucleus is gone. The chromosomes begin to condense, making them easier to pull apart. The mitotic spindles starts to form, all signs that nucleus is getting ready to break down. This is late prophase, sometimes considered pro-metaphase. Mitotic spindles begin to capture the condensed chromosomes, and the nuclear envelope begins to break down. Now we're going to take a look in the spindle anatomy, specifically in the chromosome. The point of connection in the chromosome is the centromere, and the point of attachment to the chromosome is known as kinetochore. Now, the second step is metaphase. At this point, the spindle has captured all the chromosomes, lining them up in an invisible line known as the metaphase plate. Before moving on to the next step is the spindle checkpoint. This is making sure that all chromosomes are at the metaphase plate and attached to the microtubules. The third step is anaphase. All of the pulling done in this step is done by using motor proteins. The sister chromatids separate from each other and are pulled toward opposite ends of the cell through the use of kinetochore microtubules. You can see that happening through the separation of the chromosomes in the center of the cell. Microtubules not attached to the chromosomes elongate and push apart, separating the poles and making the cell longer, giving it the H shape. The fourth and last step is telophase. The cell is nearly done dividing and starting to re-establish its normal structure. As you can see, the mitotic spindle disappears and the nucleus and the nuclear membrane reappear. The chromosomes begin to decondense and return to their stringy form. The continuation of the last step is cytokinesis. In the animal cell, actin rings is formed before the completion of mitosis can occur. The result is two identical daughter cells. In the plant cell, the only difference is the lack of elongation due to its stiff structure, thus the formation of a cell plate resulting in two plant cells.